Hello, everyone. Welcome. This webinar is now live and being recorded. We'll get going in just a moment as everyone continues to log in. You can enable closed captions today by clicking in the caption button, then the show captions. If you are new to our webinar series, welcome and thank you for joining us. Criterion Edge runs multiple informative webinars throughout the year. Today, we are presenting Pro Tips Unveiled, Expert Systematic Literature Review Strategies for EU IVDR submissions. Before we begin, I would like to mention a few housekeeping items. If you experience any technical difficulties, please use the chat option on your Zoom menu bar to send us a message, and we'll do our best to resolve it. You can enable an audio transcription for our webinar today by clicking on the captions button and clicking show captions. A full transcript can also be enabled so that you can follow along with our presenters throughout the presentation. If you have any questions about our content today, you don't have to wait until the end. There's a Q&A option on your Zoom menu bar. Go ahead and send in your questions at any point during the presentation, and we will run through as many as we can afterward. If we don't get to your question, we will get in touch with you later by email. Throughout the webinar, we will also be sharing important resources and links in the chat for all our attendees to access, just like other related webinars or information sources. This webinar, again, is being recorded and will be available to watch on our website afterwards. You will also receive an email from us with a link to the recording and slides. Lastly, I encourage everyone here to take our short survey once the webinar ends. Your feedback helps us to create content that matters most to you. Now, let's go ahead and get started. We're glad that you joined us today for our live webinar with Dr. Sarah Chavez, who will provide key insights and guidance on the purpose and scope of the state-of-the-art and competitor systematic literature review, the approach to the searches and screening, and the considerations for data presentation based on early notified body feedback. Next slide. Dr. Sarah Chavez is the Director of IVD and Scientific Writing Services with Criterion Edge. Sarah has a Bachelor's of Science in Biochemistry and a PhD in Molecular and Cellular Biology with 25 years of laboratory bench experience and has taught at the university level for the past 15 years. She has extensive scientific writing experience in an academic environment, as well as for several large textbooks publishers. Sarah's strong background in basic research and her extensive experience with regulatory writing allows her to assist companies with IVDR readiness as part of the Criterion Edge team. We'll turn it to you, Sarah.
Thank you, Cheryl, and thank you for everybody who has joined us today for this presentation on how systematic literature review can help you in your preparations for IVDR readiness. We really hope this webinar will teach you some practical guidance on how to conduct a systematic literature review and provide some insights into how the published data is leveraged for critical IVDR required reports, including the Scientific Validity Report, or the SVR. Now, we all know companies need data, whether it's to inform your organizational strategies or to guide decision-making in terms of your research and development, but ultimately, the actual systematic literature review process really poses unique challenges to a scientific team, especially in the IVDR space. For many of you, you may not have access to a medical librarian to consult with or to design a sound search strategy with. There are also many of these regulatory compliant processes that should be in place to help you document the search, whether it's searching, screening, and also your selection activities. And then finally, it comes down to actually executing the systematic literature review. That requires people. And even with tools and technology, it is quite time intensive, and time and people may be in short supply within a company. So the bottom line is really to maintain a consistent process and to try to increase efficiency. And that can be difficult when you're dealing with resource constraints, or if you really don't even know what they're asking for. So really in today's regulatory landscape, adopting this transparent and reproducible process for literature review is absolutely essential. Oh, next slide, please. So in today's webinar, we are going to be really taking a deep dive into the currently accepted systematic literature review processes, and also helping to provide guidance for how to design and conduct a rigorous literature review, from search strategies through screening and data extraction and reporting. So I hope by the end of this webinar, you will have a better understanding of how leveraging published data can fill those knowledge gaps and also meet IVDR expectations. We want to cover why the systematic literature review is superior to a simple internet search or even a PubMed search, and then discuss some of the steps for conducting a methodologically sound literature review. And then finally, we'll end with a relevant case study scenario, which will hopefully help to illustrate how the data obtained through the systematic literature review process can really support you across multiple IVDR reports. So next slide. And here we will be just starting at the beginning. So we want to start with a description of the framework of a literature review, and then also examine how conducting a literature search is much different. So that's where I'd like to start. When we talk about a systematic literature review, first off, this is not an optional activity. It must be a clear plan that's going to document how you're going to identify, retrieve, select, appraise, and ultimately use the published data. So this whole process is really nothing new. Systematic literature review is the gold standard when you're in academia or in many professional arenas. The data from systematic literature review is ultimately what is used to develop clinical practice guidelines and recommendations, and to support many academic endeavors. So the issue here is that the same rigorous principles are now being mandated by the global regulatory authorities, and they're currently being adopted by pharmaceutical, medical device, and IVD manufacturers as best practice. The defined and methodologically sound procedure they're referring to is for the purpose of assessing your device's scientific validity, analytical performance, and clinical performance. And so the expectation is that you can then document this process to ensure transparency as well as reproducibility. Okay, next slide, please. And I'd like to point out first that there are many differences between an ad hoc search using the internet or even a PubMed search, as we'll see later. They're definitely not equivalent to one another. So the internet searches can be used and often are a good starting place if you are new to this process to help you determine the types of keywords that you should be incorporating into your search. But they should absolutely be considered ad hoc because they don't follow a predetermined planned methodology that you'll be undertaking as part of the SLR process. So ask yourself, first off, can the search results be considered comprehensive and thorough? Not really if you're talking about an internet search, because it will change from day to day. And there are algorithms that define which searches and which articles are retrieved. It's not objective. And then finally ask yourself, can a literature search actually search across multiple databases if you're just using the internet? Google Scholar can be a good help, but it's really not considered to be one of these required databases. Really, PubMed is the only major database that can be accessed on the internet without a paid subscription. But it's assumed that a systematic search is being run on a platform with access to all relevant databases. At a minimum, that should include Embase and Medline. And then third, can your search platform support flexible search terminology? I will be providing you with an example of this in our case study, but the words that you use and the search terminology that you select will have a direct impact on the total amount of data and articles that you will be screening. So it's really important to pay careful attention to the terminology that you're using. Then the other issue, and one of the major issues with internet searches, is the methodology. It's hard to really clearly document and reproduce the methodology for an internet search. So that's a definite no. When it comes to objectivity in screening and selection, that's also a major issue for the internet. You know, it's very difficult to apply the same level of rigor and objectivity when you're searching something across the internet compared to a true systematic literature review. So next slide. This is why we thought it would be really helpful for this webinar to dive into the details on the methodology of the systematic literature review process. Basically give you a preview of what they're expecting you to do. So here's the bird's eye view of the basic steps of every SLR. First off, you need to define and develop your research question, and that's going to entail having a thorough understanding of your IFU. Then you're going to move on to develop your search strategy and actually conduct the search. And then next is where you're going to screen all of the literature that was returned using predetermined exclusion and inclusion criteria. Then you'll be extracting the data. That part can be time consuming and will include a thorough review of full text articles that you've selected during your screening. And then finally, you'll be synthesizing, organizing, and analyzing all of that extracted data into various reports to ultimately answer and support the research questions. Okay, next slide, please. And here, when we want to get started on a search, I always tell people, start with your intended purpose. That will be your compass, that will be your guide. Ultimately, the intended purpose for an IVD falls into a couple major groupings. Ask yourself if your device is for the purpose of screening, diagnosing, perhaps an aid to diagnosis, if it's part of classification, prognosis, risk monitoring, or therapy selection or stratification. So you'll then ask yourself, what sources of data do I have available? Those will carry over and support multiple reports within the IVDR process. So that will include things like the scientific validity report, the analytical performance, as well as the clinical performance. But then you also want to capture the information about the state of the art. So that will be one type of search that you'll want to carefully craft, but use your intended purpose as your guide. That will support your scientific validity. But you also need data. So you need to know what types of data you're looking for, and those are the performance objectives, which will ultimately live in your analytical performance and clinical performance reports. And then finally, you need to justify the benefits and potential risks of false test results. Those will live within the scientific validity. So as a point of best practice, use strictly defined eligibility criteria. Those will include your PICO terms, your study design, and the various report characteristics. So in the next slide, I'm going to try to walk you through the basic process that we take when we first take on a new project. First off, we ask the basic questions that are aligned in IVDR Annex 1, Chapter 3. In order to establish the scientific validity and to approach the intended purpose of your device, you have to clearly describe and illustrate what you're detecting and measuring. What is your analyte? What's the function of this test? Is there specific information this test will provide? Even things as simple as whether or not the data is quantitative or qualitative can have a really big effect, because those will affect the types of performance objectives you'll be capturing. You'll be discussing what type of specimen is included within your intended purpose, as well as the intended population that you'll be covering. So ultimately, a well-defined scoping process will provide the foundation for your entire literature review. It's really important to get this first step right. And you want to make sure that your search strategy is both thoroughly documented, but also focused and well-defined. So next slide. Now we're actually searching. So when you are doing the search, you're going to need to identify certain parameters, and that's where it helps to have a clear knowledge of the product names that you're going to include within your competitor devices, as well as your PICO terms, your population, indications, comparators, and outcomes. You'll also want to determine the exclusion criteria. So for instance, you may want to exclude animal studies if you know that your particular device has a strong publication history, if there's a lot of information out there. If you're a novel device, you may not want to make that exclusion criteria. You may actually need to use some of the data from preclinical studies to support the scientific validity for a novel device. You also may want to determine certain search limits, and that can include things like the time range or even the levels of evidence. If you have a lot of data and you know that there's a robust history and a lot of articles that have been published, it may be helpful to limit yourself to high levels of evidence. So systematic literature reviews, meta-analyses. If data is more scarce, you may need to expand that time range, or you may need to make justifications about why you're considering lower levels of evidence in your final search results. So a couple of key takeaways: work closely with a medical librarian. They will help you refine this search strategy and get the search strings correct. And then also maintain a very detailed record of the results of this search. The documents that all potentially relevant articles are going to, those documents are going to be scrutinized by the notified body, and you want to make sure that you can justify your inclusion and exclusion criteria. Okay. In the next slide, we're going to be looking at screening. So here is one of the most time-intensive parts of this process. Here's where you're actually identifying the appropriate literature. You're trying to minimize selection bias. So that's where having those clearly defined inclusion and exclusion criteria are extremely helpful. So you want to make sure that you are able to support those eligibility criteria and then use those to help you make objective screening decisions. So this whole process should be transparent and reproducible, and you want to ensure that your decisions and the screening criteria you're selecting are compliant with the global regulatory standards, including those outlined in MedDev 2.7/1 Rev 4, which I will mention again later. The list of excluded references, as well as the reason for exclusion, are common and expected reporting requirements now. So I'm sure this isn't coming as a surprise to most of you on the call here, that this can be extremely time consuming. And one strategy that we've come up with to manage this challenge of screening is taking a multi-stage approach. So there are some articles that simply by reading the title and abstract, you can identify as being a very straightforward screening decision. Perhaps it's off topic or not relevant. Perhaps it's technical data or preclinical data, or maybe it's just, it's not going to provide the exact intended purpose or data relevant to your intended purpose. Those can be eliminated at the level 1. Once you get into level 2 screening, that's really where you have to take that deeper dive by looking into the full text. But by eliminating some of the obvious choices at level 1 through a title and abstract screening, it can save you quite a bit of time and allow you to really focus your time and efforts on the full text analysis at level 2. So the next step in this process is data extraction. And again, that should be prospectively planned and consistent, but it can be hard to know really what you're going to find. If you knew what you were going to find, we probably wouldn't be doing this whole process in the first place. For many IVD companies, I'm finding that the amount of data that can be retrieved through the systematic literature review process may or may not be what you expected. And it may require that you, have to be looking in additional places for the data. You may need to go beyond just Medline and Embase to be able to capture enough data to support your intended purpose. So as I mentioned earlier, in terms of that scientific validity, you would be extracting PICO data to serve, to inform, and ultimately describe the conclusions that you've made with regard to the intended purpose of your device. And you're on the lookout for both analytical data as well as clinical performance data, not only for your device, but also those devices which you consider to be your competitor. And also the state of the art. You want to describe how else would you make this particular diagnosis? What other types of options are out there? Those are alternatives, not your competitors. So at this step, we also like to point out that the next step really is that you're going to want to judge the appraisal of this data set. So that's what is here in the next slide, step 5. So in step 5, we're going to be using some sort of a standardized appraisal methodology. There is one put out by the Oxford Center for Evidence-Based Medicine. We commonly use the Global Harmonization Task Force guidance, and it's a very helpful, very robust, and well-accepted appraisal tool. We have found, though, that when you're pulling together the data extraction forms, it helps to take a little bit of that human error out of the process by utilizing some of these different computer platforms. And I know that those were some of the questions that came up in that we've received from some of you who are logged in today. So we will definitely provide some insights and some advice in terms of how to avoid those human errors and maybe automate a bit of this process during that data extraction and appraisal step. And then finally, in step 6, this is ultimately where you present and report out your screening results. This PRISMA reporting structure is a very clear systematic flow diagram, and it should document the results from each step of your systematic literature review search, screening, and review. And the documentation requirements may vary depending on who you're submitting your reports to, but they typically do include some, if not all, of the requirements that we've shown here. A PRISMA flowchart is typically part of the documentation. If you use a screening software platform such as Distiller, creating that PRISMA flowchart can be automated and is quite easy, but it can be done manually as well. So our key takeaway from this slide, really, as we're concluding, is to just state that as a general rule of thumb, your documentation needs to be sufficiently detailed that if anyone were to pick up this particular document and attempt to replicate it, they could. So details are appreciated and expected in this whole process. So now I'd like to move into sort of a case study. A lot of what we've described here is all great in theory, but what does this actually look like? What are some of the considerations you need to make? The IVDR is a significant change, specifically this language regarding the systematic literature review. So in this example here, I wanted to provide just sort of a topic area that many of you may be familiar with. So that's why I selected HIV. So suppose you have a company who's decided they're writing their PER for an HIV diagnostic test. That would fall under the Class D. So that's another reason I chose this one, because we tend to see a lot of the Class Ds who are kind of the first in line under this new regulation. And this device is designed to detect HIV in blood samples using ELISA. So the company recognizes that if you have a PER, you need a systematic literature review. So really the first step for many companies is that gap analysis, trying to figure out what information do you have that you can pull into some of these required reports. But if you don't have a full systematic literature review, it's hard to really even know where to get started. So, that thorough review of your internal documents and sort of a gap assessment is a critical first step. But if you don't have data derived from a systematic literature review, it's hard to even get started with your writing. So you want to see what information do I have already, what reports and documentations are available, and then jump right into your systematic literature review. In many cases, we are seeing companies who are conducting these SLRs proactively, even while devices are under development, because it does give you a really solid understanding of what the state of the art is. If you are unaware or if you haven't conducted this, it's nice to know what sort of information is out there already. So our strategy and our approach for this company, and the main advice we would give them, would be to help them design a targeted systematic literature review. And that would justify and establish the scientific validity of ELISA-based HIV testing, which is in alignment with their intended purpose. And that would help bring their SVR then into alignment with the new reporting requirements. So if your supporting documents really are not in alignment or not up to par, it's hard to even get started. If you don't have that documented systematic literature review, you want to get that before you move too far into the process. So I mentioned this MedDev 2.7/1 Rev 4 before, and we refer to this quite a bit because it does provide some really helpful guidance. We need to define our question, and ultimately our purpose is to establish the state of the art. We want to show that ELISA is a scientifically valid approach for the detection of HIV in blood. We want to justify the methodology and the target analytes that we're using. And we also want to establish what is state of the art for HIV testing in general. So what do we actually mean by state of the art? IVDR mentions that terms at least 20 times, but it doesn't actually define what they mean by state of the art. And that's really where we have been referring folks to this MedDev. In the MDR world, the state of the art is defined as the current state of all competitive treatment options. So applying the same basic concept to IVDR, the state of the art is really the current state of all competitive diagnostic options. So in that case, our comparators would include other ELISA-based diagnostic assays. We would have our subject IVD as well as competitive tests, which use similar methodologies. So through systematic literature review, our goal is going to be to identify published research studies, peer-reviewed studies that use ELISA to detect HIV in a manner similar to our subject device. The data from this SVR will be incorporated into the PER, so we want to ensure that the research question generates a robust and unbiased data set to support the scientific validity of the test. So in effect, our research question is basically, is ELISA a scientifically valid diagnostic methodology? So then your next step really is to figure out how to conduct that search. So here, this is where those PICO terms come back. Ask your client or ask yourself, is your population limited to certain patients? Are there some individuals for whom this test would not be a good option? Is it appropriate for both adults and children? Those are the types of questions you need to establish from the get-go before you even attempt this search. Then you want to decide whether or not the literature can support ELISA-based HIV testing. That's your intervention, is an ELISA-based assay. The comparators and controls, think about this as if ELISA-based tests were not available, if that's not an option to you, how else could you make a diagnosis? What else could you use? And then the outcome are going to be the metrics by which you use to assess risk and also compare efficacy. So how do you define success in terms of your assay? You're going to determine certain search limits and filters. Those can be including things like the dates of the search, limiting it to humans only, trying to determine how far back you want to go. So for something like HIV diagnostic testing, your literature, you can expect to go back several decades, but how far back do you need to go? Whereas with something with COVID, like COVID testing, you can really limit your search range. You're only going to be looking back to 2020, really. So that being said, more is not always better. Just because your test has been on the market for 50 years does not mean you are required to search all 50 years. that can be a major challenge and can ultimately lead to these enormous screening endeavors for some clients. It's time intensive, and while this process is meant to be thorough, you can look at what the current state of the art is, and you can look at what the current recommendations are by taking a more conservative approach. Somewhere in the 5-year to 10-year range is pretty typical. So, this step is really where you want to make sure that you've chosen the best possible search limits and filters to generate a targeted yet robust pool of studies. It's hard to know how much is enough, but you want to show that you've been thoughtful in the limits and the filters you've applied. But don't forget about the importance of where you're searching. And that's what I wanted to illustrate here in this next slide, where I want to show really a side-by-side comparison of an internet search versus an advanced search platform like Embase or Medline. So next slide, please. And when we conduct the search, you can see here, I pointed out earlier that an internet search or even a PubMed search, and trust me, I love PubMed, I use PubMed all the time, but it's not the same as an advanced search platform such as the one that we use, which is ProQuest Dialog. ProQuest Dialog can search simultaneously across both Embase and Medline, as well as additional databases. Compared to an internet search, even if it is PubMed, it's going to be limited in scope. So as you can see here on the first table on the left, these are the results of a typical PubMed search. If you just search for HIV test diagnostics, you're getting over 6,000 hits. But if you switch up the order and say diagnostics test, you drop that down to 700 articles. If you take away the plural and instead of typing diagnostics, plural, you just type diagnostic, now you've doubled your search results. If you try to limit that to PCR tests, you get about 500. If you look for ELISA, it's 1,200. The different combinations are all over the place, and they seem quite arbitrary because really you don't have the same ability to craft these search strings precisely. So it really is a huge difference. The PubMed syntax is quite limited, and it doesn't allow you to really drill down on a research question. So, being able to search across Embase or Medline, while the upfront cost of investing in these platforms or engaging with a librarian seems like a lot, it is going to save you hours and hours when it comes time to screen these. So as you can see here on the right, when you run the same type of search, you're crafting much more detailed search strings. And this is even before you've applied too terribly many limits. This is quite a simple search. Depending on how detailed your device is or how precise your intended purpose is, you can adapt and adjust these search strings to be able to allow you to identify the most appropriate combination to give you articles that you will then go on and move on to screen. So, screening is going to take you the most time. So doing this search correctly is going to save you a lot of time and a lot of headaches moving forward. So now let's move on to the screening. We know that you have to document this process. So having a way to organize this information is key. And I just sort of copied just some basic ideas of the types of things and the types of ways that you can document this. Now, perhaps for your purpose, it comes down to the publication year. Maybe you can exclude some of the data as they're just beyond the 5-year scope. Maybe you can select things like limiting to only meta-analyses or randomized control trials, or peer-reviewed literature. If you decide you want to not include preclinical data, you can use that as a screening criteria. So the study type can actually allow you to eliminate some of these articles from the get-go. You may say a conference abstract is a lower level of evidence, so I want to exclude those. Or I want to not include preclinical studies. There are also ways to incorporate these limits for study types into the search strings, so that can help you with this screening process as well. When it comes time to actually start to identify data in these articles, we want to get to the point where, as you're screening it, you eliminate those easy ones first. So if you're limiting it to 5 years, English language, peer-reviewed literature, and perhaps you want to limit your sample size to any study over 500 patients. Or you want to limit to EU populations if you're submitting under the IVDR. A lot of times you can do that in just a screening of the title and the abstract. But a full text review is going to help you really assess the types of HIV screening tests that are used. And I'll warn you, what I'm finding with many of these projects are that many of these articles will absolutely use the tests that you're looking for data on. They'll use them in clinical practice, but they may not provide the details. If the focus of the article is really to show something else, to show, we tested patients and then did this, offered them this treatment, that's very common. So yes, they'll report on testing, but that's not the focus of the article. And so there may not actually be extractable data within that article with regards to the accuracy or the sensitivity or the specificity of the assays. Similarly, you may come across some articles which are method comparison papers, where they may compare the accuracy of ELISA-based testing to PCR testing. Those are going to be that analytical data that you're on the lookout for. But clinical data has been hard to come by for many devices in the IVD space. So I can warn you that, the analytical data you will likely encounter in these method comparison papers. The clinical data can be challenging, and that's really where designing these searches to capture any article which happens to publish on the accuracy of the test in a real world patient population, in a true clinical setting. That's what you want to be on the lookout for. So next, I'd like to give you just sort of an example of the types and approaches for data extraction. So this is really the goal of the systematic literature review process. You want to identify high quality studies that have extractable data that allow you to establish the scientific validity and the acceptance criteria by which you will measure your device. So for the SVR in particular, this will include information about your competitors, but you should also take into account the target of the different ELISA-based assays. Sometimes they don't use the same antigen or the same antibody combinations. Different ELISAs can use different targets. And so it's important to consider that and to document that within your competitive landscape, because it could explain why some of your competitors may experience a false positive or some other risk of incorrect results that you don't have, or it can give you insights into why you may see discrepancies or a lack of concordance between one competitor and another. Those are the types of things you want to analyze and assess within the competitive landscape. For both the SVR as well as the CPR and the APR, you're looking for sensitivity and specificity data, but analytical sensitivity and specificity will be assessing things like an actual quantifiable range if this test is a quantitative ELISA. But maybe you have a qualitative ELISA, so you may not expect to see analytical sensitivity. The other thing to ask yourself is there a reference standard? Is there an established international standard that is being used to quantify these assays? If that is changed, which is the case for many devices, over time, the standards may change and improve. That would be something that you would want to capture, because data collected using an old method of standardization or an old reference standard may not be the same as the newer data. So be on the lookout for the reference standards, the cutoff values. I have noticed quite a bit that the cutoffs are often different than what is recommended by the manufacturer. So pay close attention to that, and also the reference intervals for different populations. So it's not just collecting sensitivity is this, specificity is this, done. You want to analyze it in the context of what were these studies conducted using the same intended purpose that I'm claiming. So ultimately, this is why it's so important to record all of these details about the assays and to allow you to make those direct comparisons in your analysis. So after you've then extracted all of these data, you should be prepared to document the strategy and the approach for the process itself. And so our last step really is in the process of systematic literature review is to describe how you came about your final data set. If you identify that there is a potential outlier in your data set, you can include it, but you can also make note of that within the data extraction table. But the Prisma flowchart is providing you that high-level strategy and overview that tells the reviewers how and why you selected the articles that you're including, that you're using to develop your acceptance criteria. These are the data ultimately that you're using to inform your final conclusions. So if you are going to exclude a particular article, you should be prepared to justify that exclusion. And similarly, you wouldn't want to include ad hoc articles to support your data or lower levels of evidence. You want to ensure that the appraisals as well as the process itself supports and can help you establish your confidence that the studies you've identified and the criteria you've selected establish and support the scientific validity of ELISA-based HIV diagnostic testing. And really, this is the purpose behind them requiring this exercise now. It's ensuring that we have IVDR compliance, but it's also ensuring that the conclusions we're drawing are derived from a reproducible and well-documented objective process. So in the end, the company needs to start with this systematic literature review, and that will support the development of their supporting documents, their SPR, CPR, APR, and then ultimately the PER. And by doing this process first and doing it well, you can be assured that your PER and the data within that document are aligned with the new IVDR guidelines. So I hope that this case study, while it is specific to one type of device, I hope it helps to illustrate how systematic literature review can be used and refined to ensure that you have these IVDR compliant technical documents. So just to review, my primary objective in today's webinar is really just to illustrate what the notified body expects in terms of the documentation of this systematic literature review process and why it's now the required standard for all notified bodies and global regulatory authorities. We wanted to show, in theory, how to conduct a systematic literature review, all the way from scoping it correctly through data extraction and reporting. And then we wanted to illustrate how to implement these required steps within your own organization. That can be challenging, and sometimes there are specific nuances with your particular device that you may not find an exact guidance that really fits your device. And then finally, we wanted to illustrate that this data can be really helpful in terms of supporting your organization's IVDR readiness, but can also be useful for your research and development teams ultimately. So it all comes down to what the notified body is expecting to see out of your submission. They want to see that you've chosen appropriate and clinically relevant performance objectives in the context and considering the state of the art. That's your SPR. They want to see a list and specification of the parameters by which you determined acceptability of your benefit risk ratio of your subject device. Those are your acceptance criteria. They want to see that you've clearly organized all of these data sets, and that can be challenging. And that can include not only your manufacturer held data, but also clinical registry data and these published studies, which we've described today. Those will support your CPR and your APR for your subject device. And then finally, you want to illustrate the acceptability of the risk-benefit ratio as supported by the data. That's really the goal of the PER. But don't forget, you need to document the process by which all of these data were collected.
Well, thank you so much, Sarah, for all the information and guidance. This has been an excellent discussion. We'll now turn to our Q&A portion. We have taken some time to review the questions that we received from you beforehand. Thank you for your submissions. Now, if you have questions, since the presentation is done, please go ahead and send them in as well using the Q&A button, and we'll run through as many additional questions as we can. While your questions are coming in, I want to remind you all that you can reach out to us at consult@criterionedge.com to schedule some time to talk with our specialists. We have recently worked on several IVDR submissions and are available to chat with you about your current or upcoming project needs. And you can also book a meeting time with us via the email that you'll receive following this session today. So we'll turn to question 1. Sarah, shall we get started? Where is the best place to start searching for literature?
Yes, so we get this question a lot. We will often get approached by folks who've attempted the PubMed search and then tell us, I have 10,000 articles. This cannot happen. Like this, surely this isn't what they're expecting. And no, that's not what they're expecting. So the best place to search is going to be starting with Medline and Embase. You know, people either fall into two camps. They either have too much data or not enough data. That seems to, nobody comes at these projects and say, I have exactly the right amount of data, you know? So as you're getting started, start at a minimum with Medline and Embase. And then if you determine I have way more data than I want to attempt to extract or that a typical notified body wants to review, then you can scale it back using those filters.
Great, thank you. We'll turn to the next question then. Is there an electronic system that manages this process or type of review to aid efficiency for companies?
Yes, so there are several different options out there. The one that our team uses, first off, we use Distiller to help us with the review process. It's basically a software that allows you to do everything from the screening to the extraction to the Prisma. It's sort of like a database that maintains a record of these projects. That can be extremely helpful if you want to, if you have similar device families and you want to start archiving these projects, or if you want to go back to a previous data set for like a, you know, doing an update or a gap analysis or something. You also want to try to use something like a search platform like ProQuest Dialog. That is going to be really helpful with that literature search. You can absolutely search Medline and Embase independently, but the benefit of something like a ProQuest Dialog is that you can actually search across both platforms simultaneously using the same crafted search, and it can save you a lot of time and a lot of deduplication time.
Thank you, Sarah, for answering both of those questions for us. We'll turn to question 3. Are there any specific differences related to IVDR searches in comparison to MDR literature searches?
We have gotten this question quite a bit too. You know, when it comes to the IVDR, I think everybody's looking for hints about what exactly is going to happen. So in terms of the IVDR, the main difference that I've found is going to be in terms of that analyte. So we really want to make sure that that analyte is captured and that we're justifying the choice of the analyte in our search strings.
Wonderful, thank you. How does one formalize the search for competitors?
This can also be really challenging. You know, when we have the competitor search, I have seen folks who have tried to search in just the title and the abstract for the competitors, but any of you who've sort of started to dig into this process may have found they don't typically mention, I'm testing this assay versus that assay. That's a method comparison paper, and those are typically analytical performance. But if you want to dig in and find clinical data on one of your competitors, you would want to search all fields and you would want to use the device name or you would also want to use, sort of the intended purpose for which that device is being used as kind of limits. So we devise searches. We have a unique standalone competitor search. So we actually run independent searches for the subject device, the state of the art, the guidelines, and then the competitors. And the competitor search will incorporate the names of the devices as well as the intended purpose. And it will search across all fields to hopefully capture the articles which use that device, even if it's buried within their methods section, you can find it.
Excellent, thank you. And question 5, are other sources than peer-reviewed articles also allowed?
Yes, with an asterisk. So for instance, if let's say you have identified a conference abstract or a poster, and it has competitor data or the IFU from your competitor, those can be mentioned, but those would not be part of that final data set that you're using to establish your acceptance criteria. Because what the IVDR is ultimately intending this to serve is to show that, yeah, it's great if in your lab this particular device performs with a sensitivity of 99%. But when it gets out into the real world, in real people's hands, does it still perform the same? So we are very careful to make it clear which articles are being used to derive the acceptance criteria, which are these peer-reviewed, high level of evidence articles that support the competitive landscape. And then if there are these non-peer-reviewed articles or book chapters, things like that, you can include those in your background section to support the state of the art, but they wouldn't contribute to the data set.
Thank you. And we have just 3 remaining questions here. The next question is, does the literature review search strategy change as the device risk classification goes from A to D?
This is a good question. And I think it's actually the opposite of what a lot of people think. In my experience, the lower risk class devices are harder to find than the higher risk, and I guess in retrospect, it makes sense because if it's a high risk device, there tends to be a lot of clinical data published on it. So a lot of these lower device risk classification, or even if it's more of like a general device, or if it has multiple broad intended purposes, that's really where it becomes extremely challenging to get the search strategy refined enough that you can capture that data, much less to find performance data on that device. So you definitely want to incorporate your competitors' names into the search for your device or your competitive landscape. But you may need to get a little bit creative in terms of the filters and the strings. You may need to play around with the time that it takes if it's one of these sort of newer or a well-established device. If in doubt, always start with the guidelines. The guidelines will kind of show you where the most recent data is and where the most recent trends are, especially if you've got one of these sort of lower risk class devices.
Wonderful. Thank you, Sarah. And before we get to our next question, we did have one just post here that I'd like to be sure you speak
Oh, sure.
to. If you need to perform the screening manually without an informatics program like Distiller, how would the SLR be performed? Do you receive a kind of folder with all the articles retrieved by the librarian?
Yes. So if you are not using a specialized program, the typical methodology, many people just use an Excel document to help to document the process. So you would run the search. You would receive a list of articles in a Word format, but you can also receive it in an RIS format, which you can then import into a reference management system like an EndNote or something. And then you would conduct the screening and document it within sort of a spreadsheet format. So the trick there is that you would then need to manually archive all of this information and document this information and manually build your Prisma. You can also, the other challenge is also how do you maintain all of the the organization for all of these PDFs. So I typically, if someone is not using Distiller, I recommend a combination of Excel and EndNote. And that can usually get you a system that the majority of companies who don't use Distiller regularly could easily access and retain for their long-term documentation. So yeah, at a minimum, if I were attempting this without specialized programs, I would want EndNote and Excel.
Thank you, Sarah. And we have time for just 2 more questions. Here we are with what is the difference between state-of-the-art SOTA and SVR, Scientific Validity Report, in terms of structure?
Honestly, they're quite similar. Really, the main place and the main difference for those folks in the MDR space, they're very familiar with the state of the art and that competitive landscape and acceptance criteria and all of that. The main place that I've found where the scientific validity report kind of expounds upon that is that we have to take the extra added step of justifying the association between the analyte and the clinical condition. So we do spend a little bit more time describing how the device works, the basic science underlying the device. And that really would be in a different section outside of the SOTA in a typical medical device CER. But here in the SVR, you really want to show not only that this is the right test to use, but how does that test work from a science perspective?
Wonderful. Thank you, Sarah. And our last question today, question 8. Is it necessary to conduct a search in the CDSR Cochrane website when you already searched the 3 clinical trials websites?
No, it's not necessary, but I would suggest taking a peek, because what I found is that those Cochrane reviews have a lot of really helpful background information, as well as they've typically consulted the guidances. So they can help you support your guidelines as well as some of the basic scientific validity steps that you're going to take. So you tend to see quite a bit of overlap, but those reviews can be extremely helpful in terms of, the background information you need to write these state of the art.
Wonderful. Thank you so much, Sarah. Well, if you have enjoyed our webinar today, you may want to sign up to be notified of additional upcoming webinars produced by Criterion Edge. You can do so by clicking on the link that we've just provided in the Q&A section of your Zoom window. If you still have questions on the content we discussed today, or if you'd like to learn more about our services, you can schedule a free appointment with our specialists. Our contact information is consult@criterionedge.com, which is what you see here on your screen. This webinar, again, is recorded and will be available soon to watch on our website, and it will be sent out to you with a link and an email with the slides and all of these related announcements. As a reminder, when you exit the webinar, please take our short survey and let us know how you enjoyed today's talk and what other topics you would like us to cover in the future. Many thanks to Dr. Sarah Chavez, our Director of IVD and Scientific Writing Services here at Criterion Edge, for the excellent presentation, and to all of you for attending and for your questions. We hope this presentation gave you more insight into how to perform strong foundational systematic literature reviews for your IVDR CE mark submissions. Have a great rest of your day, and we'll see you next time.